must be knowing about biogeochemical cycle. Biogeochemical cycle. This biogeochemical cycle, there are various cycles: carbon cycle, nitrogen cycle, phosphorus cycle, water cycle. In this whole cycle, microorganisms play an important role. It was discussed. It is an example of food chain. We know that in the presence of light, green plant carry out photosynthesis, convert inorganic carbon dioxide into organic form. Then these various stages are known as trophic level. This is trophic level first. They are autotrophs, photosynthetic organisms. This is trophic level 2. They are primary consumers. Trophic level 3, secondary consumer, tertiary consumer, likewise. But when all die, they are decomposed by microorganisms, where several type of bacteria, fungi, actinomycetes, all type of bacteria play an important role in decomposed body. All carbon, sulfur, phosphorus, they go in the air. Again carbon undergo photosynthesis, again it converts into organs. So this cycle goes on. So there is a major role of these microorganisms. Next. We know that what is microbes and microbiology. You all are known about this stuff, that microbes include viruses, prokaryotes, two types of prokaryotes, bacteria and archaebacteria. These are also known as eubacteria and these are archaebacteria. These are also known as extremophiles. ઘણીવાર પિક્ચરમાં આપણે જૂના પિક્ચર 80s ના પિક્ચર જુઓ ને, ત્યારે હીરો અને હિરોઈન જાય મેળામાં, એના બે બાળકો હોય, બે બાળકો ખોવાઈ જાય, બરાબર? અમિતાભ બચ્ચનનું પિક્ચર છે. એક પછી ગુંડો બને ને એક બને પોલીસ, પછી પાછા ભેગા થાય. And this is how picture goes on. These are this type of brothers. When life originated, there are only one type of bacteria, but as they thriving in different environmental conditions, some develop as bacteria, eubacteria, which can grow at normal temperature. And archaebacteria, for example, methanogens, halophiles, thermoacidophiles, they found at extreme conditions. So these bacteria comes under prokaryotes and eukaryotes, algae, fungi and protozoa. Next, we will go through these are how can microbes can be classified, that you know very well, that fungi, protozoa, algae, bacteria, archaea and small animals. Next. Now this is the traditional Whittaker's classification system that is also been, already it is discussed. Based on their nutritional requirement, this classification is given, and there are plantae, animalia, fungi, protista, archaea and bacteria. These are multicellular again, but mode of action of this nutrient uptake is different. Next. Now, how to cultivate these microbes? There are 5 I you are supposed to remember when you want to cultivate microbes. Inoculation, incubation, isolation, inspection and identifications. These are 4 ways of getting to study that microorganisms. What we are doing in inoculation, because each and every microbe has its particular requirement. So based on the requirement, we are preparing media and we are introducing a sample on that media. Then at a particular temperature, we are incubating. Then next day we are observing for is there any growth is there or not. Then we are going for identification. Next. And you can have this type of mixed population next day, and based on you can see these are various colonies. Rajkot juo to there are various colonies. No, Rajkot there are thousands of colonies, and in colony ma there are certain colonies are specific colony. કે અમુક કાસ્ટના લોકો જ રહેતા હોય, અમુક મિક્સ કોલોની હોય, એને મિક્સ કલ્ચર કહેવાય. Fine. And see, this is pure culture. They have one type of microorganisms. Many times you can see these are overlapping. Many more than one colonies are merged together. So these are few colonies you are getting when you inoculate very diluted soil sample on the nutrient medium. Next. So these are some example of microorganisms that you can isolate at the end of this all 5I process. Next. Now growth. Growth is the typical characteristic of bacteria. Bacteria with 60-minute generation time. Generation time and growth rate, there are two popular terms in microbiology. Generation time means a time required by a bacterium to divide into two. And growth rate, number of division per hour. Say for example, a bacteria have 20-minute generation time. I won't answer. Generation time 20, again I am defining. Generation time means time required by a bacterium to divide into two. એક બેક્ટેરિયાને એકમાંથી બે થવા માટે જે સમય લાગે એને કહેવાય generation time. એક કલાકમાં જેટલી generation થાય એને કહેવાય growth rate. Bacteria with 20-minute generation time, what will be the growth rate? What will be the growth rate? કેટલી generation એક કલાકમાં થાય? એક તો number of cell થાય. Three generation. At the end of 3 generation, you have 8 bacteria, isn't it? Now, here we are talking about not 20 minutes, but 60 minute generation time. For example, एक bacteria 60 minute માં એક માંથી બે થાય છે. So, one bacterium cell could produce these many cells in a day. એક bacteria time T0 એ divide થવાનું ચાલુ થાય 24 કલાક પછી આટલા bacteria થાય. Bacteria with generation time 60 minutes I am talking about. But if it is 60 minutes, then you can imagine. 20 minutes, then you can imagine. Viruses now smaller than the bacteria. Viruses, since they can produce several during each replication, range from 15 to 20 to 100, even 1000, they can easily produce several billions of viral particle baby viruses within 24 hours. Next. So bacteria grow exponentially. That was already discussed. That it divides 1 into 2, 2 to 4, 4 to 8, 8 to 16, 16 to 34, 34 to 60, something like that. It is arithmetic progression. Fine. So this is arithmetic progression unless and until you keep on giving fresh nutrient. You keep on giving fresh nutrient. If there is scarcity of nutrition, your bacteria will convert into spore. Next. I would like to show you some videos of how bacteria divide. It's a 1-minute video. You can see here. See carefully how bacteria divide.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Fine.
Bacteria reproduce very simply and rapidly by doubling.
It's the same, but you can see again. The green color is, these are bacterium. You can see now they are dividing. Thank God, the rate at which it is given, they are not multiplied. Otherwise, you can see there are always bacteria on this. Fine. Thanks. Now, this cell division is not a simple process. As we know that this bacteria within 20 minutes is divided into two. It's a long process. Because we know that there are 4 macromolecules: carbohydrate, lipid, protein, and nucleic acid. These all macromolecules are made up of micromolecules. They are basic monomers, and there are various processes are included before bacteria divide. Cell, first of all, when bacteria divide, before cell divide, DNA, it is again segregated to 2 differential cells. Then inward invagination of bacterial cytoplasmic membrane take place, and bacteria divide. I will show you at the end of my lecture. I will show you one animation that how many type of processes are take place when before bacteria divide. Next. Cell size. We don't have internet, no? We know that cell size also already been discussed, that microbiology. We know that why it was called microbiology, because measurement is in micron. Fine. 1 meter, how many micrometer? 10⁻⁶. So this is the range of scale, various type of microorganisms. Next, I'll show you. You can compare this bacterium from an adult human being to an atom, and these are the measurement scale. So microbiologists are working at very minute level. There are two fascinating branch of research, you can say. One is astronaut, that dealing with big things, and one are microbiologists that dealing with very minute object. Next. Now, next. Why study microbiology? Because I am here to explain that why you should choose a field of microbiology as your career. Right. So, microbes were first on this earth. Microbes created the biosphere that allow multicellular organisms to evolve. Organisms હતા એટલે આપણે છીએ. ન હોત તો આપણે અહીંયા ઇન્સ્પાયર પ્રોગ્રામમાં બેઠા ના હોત. Fine. Multicellular organisms evolved from microorganisms. More than 50% of the biomass on the earth is comprised of microorganisms. If there is no biomass, there is no food, and higher organisms cannot survive. Microorganisms will be on earth forever. આપણે હસું કે નહીં હોય, આ મિત્રો તો અહીંયા રહેવાના છે. Fine. Next. Microbes, you know that there are many types of microbes. Normal microbes, opportunistic pathogens, and pathogens. You may ask question that did you always have them? Yes, they are always with us. Are they everywhere on your body? Yes, they are everywhere on the body. And are normal flora ever harmful? They are sometimes opportunistic pathogen, part-time bad boys. And pathogens, full-time bad boys. Opportunistic pathogen, whenever they get unfavorable condition, they start causing disease. Thanks. Microbial associations. We have many type of relations in society, isn't it? હોય ને ઘણા પ્રકારના રિલેશન્સ હોય. અમુક રિલેશન્સ ઓપોર્ચ્યુનિસ્ટિક હોય, કામ પૂરું થઈ જાય એટલે ટાટા સી યુ બાય બાય. અમુક લોંગ ટર્મ રિલેશન હોય, we don't have any expectation. So microbes also have many type of relations. Symbiotic, non-symbiotic. What happens in symbiotic relations? Organisms live in close nutritional relationship required by both organisms. Both partners are required for their existence, while in non-symbiotic organisms are free living. Relationships are not required for survival. If you see, this symbiotic can be further categorized under three relationships: mutualism, commensalism, and parasitism. For example, obligatory dependent, both members are benefited. For example, mycorrhiza and higher plant. They are example of mutualistic relationship. Commensalism. Commensals benefit others, members, not harmful. Skin flora. Fine. There are many type of microorganisms, but normal flora are not harmful to us. Parasitism. They are dependent, gets benefit, but they damage host, many pathogens. Likewise, you can see non-symbiotic relations, synergism and antagonism. Members cooperate and share nutritional requirement. Your flora in the gut, for example. There are pathogens are there, even normal flora are there. Pathogens might bear witness, normal flora might bear witness. Fine. Independently they are sharing nutrient, but they are not harming each other. Antagonism. Some members inhibited or destroyed by others. For example, antibiotic producing microorganisms, or some organisms that produce some chemicals that inhibit other microorganisms. And this provision is to survive under unfavorable condition. I will give one example. તમે કોઈ પણ પ્લાન્ટ લે, ચોમાસામાં પ્લાન્ટ ઉઠાડો એટલે નીચેથી રૂટ હોય, રૂટ. Fine. That area is known as rhizosphere. If you take the plant in summer and in monsoon, and if you go for microbial diversity, in monsoon you will find hundreds of species in rhizosphere. But in summer, there are less species because nutrient is not available, nutrient is less. So the flora of that rhizosphere start competing. And those who produce such type of antimicrobial chemicals, they survive. Say for example, in monsoon there were 10 species. In summer you will hardly find 1 or 2 species. Fine. Next. Impact of infectious disease. We know that if you see the average lifespan, microbes indirectly affecting your lifespan. If you see in Bronze Age, it was 26 years average lifespan, because many people were suffering. Even let me tell you, 20-25 વર્ષ પહેલા વિચારો કે ઘણીવાર રૂરલ એરિયામાંથી કોઈ આવતા હશે તો ખ્યાલ હશે કે ભાઈને મીઠો પેશાબ થઈ ગયો છે, ભાઈનું વજન ઘટતું જાય છે, પણ એને ખબર ન હોય કે એને ડાયાબિટીસ થયો છે. વજન ઘટવામાં ઉપર સીધા આવી જાય. Fine. Many times people are suffering from arthritis in rural area. They don't take medicine. So if you see in that particular range, it was 26 years. Hey, in Middle Europe around this time it was 30 years. Early 20th century it was 50 to 64 years. And now world average 67 to 0.2 years. USA 78 approximately. The Europeans live and આપણા હરિયાણાના લોકો. Fine. છાસ ચાલુ કરી છે ને પીવાની. Credit goes to microorganisms because many people were dying due to several diseases. Next. Now, worldwide infection of disease that affect the health science. You can see the increasing number of drug-resistant strain, including nosocomial and communal acquired microorganisms. I don't see એક પણ તમારામાં એવું ન હોય કે અત્યાર સુધી મેડિસિન ન લીધી હોય. છે કોઈ રેજ યોર હેન્ડ. અત્યાર સુધી એન્ટીબાયોટિક્સ ન લીધી હોય એવું કોઈ છે? No. We all are taking medicine directly or indirectly. Fine. So there are various type of disease that organisms that have developed against several diseases. For example, methicillin-resistant Staphylococcus aureus, vancomycin-resistant Enterococcus, vancomycin-resistant Staphylococcus aureus, multi-drug-resistant tuberculosis, increasing number of emerging diseases like SARS, AIDS, and HCV, hepatitis C. About 70 lakhs people die each year from food poisoning worldwide. Can you imagine? Food poisoning, and food poisoning nothing but indirectly it is microbial infection. Approximately 750 lakhs people suffer from non-fatal food poisoning. It is not fatal, but they indirectly due to these microorganisms. Next. Now, let me give you 2 examples, HIV and HCV. During my US visit, I worked on these 2 areas, HIV and HCV. See, the people are suffering from HIV global. This is the global prevalence of HIV infection. North America, 1.2 million people are suffering from HIV. Let me see here, Southeast Asia, 4.2 million people. I think this is the maximum number, isn't it? These people are suffering from HIV. Africa, yes. Next. Now, I'll give some statistic of HIV. You will surprise. More than 5.6 million people now live with HIV and AIDS. 3.4 million of them are under age of 15. In 2011, I am showing data of 2011, an estimated 2.5 million people were newly infected with HIV. You never know what is the source of infection. You are infected. 330,000 were under the age of 15. Every day, nearly 7,000 people contract HIV, nearly 300 every hour, 50 every minute, and 8 every second. Lecture પૂરું થશે ત્યાં સુધી કેટલા ઇન્ફેક્શન લાગી ગયા હશે HIV ના? Worldwide I am talking about. The population is 700 crore. So these are real statistics. In 2011, 1.7 million people died from AIDS. 230,000 of them were under the age of 15. Since the beginning of the epidemic, more than 60 million people have contracted HIV and nearly 30 million have died of HIV-related disease. Because once you are infected with HIV, other opportunistic pathogens also start playing their role. Fine. You must be knowing HIV is nothing but it weakens the immunity system, and all other pathogens who are working, they attack on the body. Next. So, For this is HIV. Now HCV, hepatitis C virus. In US it is considered dangerous than HIV. In US because they know. In India we don't know about this infection. Every person among 10 people, everyone in the group of 10 is infected with HCV. We don't know about this infection. So this is the global prevalence of hepatitis C, because we know that hepatitis, various type of hepatitis are there, but this is the dangerous hepatitis C, and there is no vaccine for this hepatitis C. 170 million people nowadays are estimated suffering from HCV. Next. If you see, there are various type of hepatitis A virus, hepatitis B virus, hepatitis C virus, D and E virus. You can see there are various modes of their transmission. For example, hepatitis A, contaminated water, food. So these are the normal water, food, these are the mode of transmission, but there is no vaccine for HCV, hepatitis C. So if you are infected, you have to be very careful. Next. And how this hepatitis C progress? Say for example, 100 people are infected with HCV. Out of 100 people infected with hepatitis C, 75 to 85 will develop chronic infection. 15% may recover. Now, if you consider this as 100%, out of them 60 to 70% will develop chronic liver disease. If you consider this as 100%, 5 to 20% will develop cirrhosis, and 1 to 5% will die of cirrhosis or liver cancer. Next. So, these are, I have given 2 examples, but there are many examples which are microbial disease. Now, infection dose, what is ID?